Imagine trying to fix like a single typo on page 50 of a massive encyclopedic book.
Right.
But your only tool is this, heavy-duty pair of gardening shears. You slice the entire page in half, right through the text, just to remove one bad word.
Which is incredibly destructive.
Exactly. And then you hand those two torn, jagged halves to a blindfolded librarian who only has a roll of cheap duct tape. And you cross your fingers that they tape it back together without covering up any of the words.
It's a chaotic approach.
It really is. But that chaotic, heavy-handed approach, that is essentially what we have been doing to human DNA for the past decade. Welcome to the Deep Dive. Today we are tearing up that old paradigm entirely.
We really are. We're moving past the brute force of, molecular scissors and entering an entirely new era of genetic precision.
Right. We are talking about rewriting the very letters of our DNA,
Right.
one single atom at a time, without ever actually breaking the spine of the book.
And it is a profound paradigm shift. For years, the scientific community has been completely captivated by the sheer power of just being able to cut the genome.
Because CRISPR was revolutionary.
Oh, absolutely. But cutting is traumatic. Now we are stepping away from the destructive force of double-strand breaks. We're stepping into the realm of ultra-precise chemical modification. I mean, it's no longer about cutting and pasting. It is about erasing and rewriting.
And to explore how we are actually doing this, we have pulled together an incredibly dense, just fascinating stack of sources for you today.
It's a really diverse stack, too.
Yeah, if you are a curious learner, maybe a clinician wondering what tools you'll be using in 5 years, or a business strategist tracking where the billions of dollars in biotech capital are currently flowing, you are in the right place.
Definitely.
We're pulling from the latest clinical pipeline reports straight out of cutting-edge biotech companies, places like Beam Therapeutics, Verve Therapeutics, Correct Sequence.
And we are digging into some seriously heavy biochemical research, too, detailing the directed evolution of entirely new synthetic enzymes.
Which is mind-blowing. We're also looking at some incredibly specific physiological studies on how our own bodies naturally try to edit RNA, focusing on this obscure but vital enzyme called ARCD1.
Yeah, that 2001 study is fascinating.
Right. And perhaps most mind-bending of all, we have a recent paper on how scientists are literally using quantum computers to simulate atomic-level genetic edits to build better drugs.
That is quite the stack. But, what connects a quantum computing simulation to a rat hepatoma study from 2001 and then to a modern clinical trial for high cholesterol is this relentless pursuit of precision.
Right.
They all point to this transformative moment in biotechnology where we stop acting like genetic lumberjacks and start acting like atomic watchmakers.
I love that. Genetic lumberjacks. So our mission today, for you, the listener, is to unpack the entire landscape of this technology, which the scientific community calls base editing.
Base editing.
Right. We want to understand the underlying mechanism, the mind-blowing subatomic chemistry, the therapeutic value, and the real business realities of it. I mean, how does a quantum simulation of an enzyme eventually translate into a lipid nanoparticle injected into a patient's arm to cure a disease?
It's a wild journey.
It is. Okay, so let's unpack this. Before we can truly appreciate how base editing works, we need to establish exactly why the original CRISPR-Cas, the world-famous CRISPR-Cas9 system, just wasn't good enough for everything.
Right. To appreciate the molecular pencil, you have to fully understand the collateral damage caused by the scissors.
Okay, break that down for me.
So, classical CRISPR-Cas9 operates through a highly specific sequence. It uses a guide RNA to recognize a target sequence in the genome. Then, the Cas9 protein acts as an endonuclease.
Meaning it cuts.
Exactly. It physically cleaves the phosphodiester backbone of both strands of the DNA double helix. It snaps the DNA right in two, and at that point, the CRISPR system's job is completely done.
Wait, it just leaves?
It just leaves. It leaves the scene of the crime and relies entirely on the host cell's natural machinery to repair the damage.
Right. And that repair process is the blindfolded librarian with the duct tape that I mentioned earlier. When a cell senses a double-strand break, it basically panics, right?
Total panic.
It uses this primary repair pathway called non-homologous end joining, or NHEJ. And from what I understand from the sources, NHEJ is fast, but it is incredibly sloppy.
Sloppy is the exact right word. Because the ends of the DNA have been severed, they are exposed to degradation by cellular exonucleases before they can even be rejoined.
So parts of the code literally just float away or get chewed up.
Exactly. By the time NHEJ glues them back together, it almost always drops a few letters out or accidentally jams a few extra letters in. In genetics, we call these indels insertions or deletions.
So if you were trying to simply knock out a gene, like just break it so it stops functioning entirely, NHEJ is great.
Oh, it's perfect for that. You cut it, the cell ruins the spelling trying to fix it, and the gene is effectively destroyed. But if you are trying to correct a really subtle mutation, introducing random indels is the absolute last thing you want to do.
Right. And it's not just about tiny local typos at the cut site, is it? I was reading about the broader cellular response. If you unleash classical CRISPR to make, I don't know, thousands of double-strand breaks in a patient's cells, the whole system can basically go into shock.
Absolutely. When you introduce double-strand breaks or DSBs, you are risking massive, catastrophic chromosomal rearrangements.
Like what?
Well, imagine cutting two separate chromosomes at the exact same time. The cell's repair machinery might accidentally take the top half of chromosome 4 and glue it to the bottom half of chromosome 9.
Oh, wow. That sounds bad.
It's terrible. That is called a chromosomal translocation, and it is a hallmark of many severe cancers. Furthermore, DSBs are just highly toxic to cells in general.
Because it triggers the alarms.
Exactly. When a cell detects a broken chromosome, it activates powerful damage response pathways, most notably involving the p53 protein.
Ah, P53.
Yeah.
The guardian of the genome.
Right. If p53 senses too much DNA damage, it halts cell division entirely and triggers apoptosis, which is programmed cell death.
It just kills the cell to save the organism.
Yeah. So if you are a clinician trying to cure a delicate genetic liver disease, creating thousands of DSBs in a patient's liver is incredibly risky. You might cure the mutation in some cells, but kill a large portion of the organ in the process, or worse.
Induces secondary malignancy, like cancer.
Precisely.
Which perfectly sets the stage for why base editing is such a massive leap forward. If classical CRISPR is a pair of scissors causing all this cellular panic, base editing is a pencil and an eraser.
A very tiny pencil.
Right. You don't cut the page. You don't trigger the p53 alarm system. You just erase a single A and write in a G. And this matters on a fundamental level because of the actual nature of the diseases we're trying to cure, doesn't it?
Yes, it really does. If we pull the data from the ClinVar database, which is this massive NIH archive that catalogs human genetic variants and their relationship to human health, the numbers are striking.
Okay, lay it on me.
Around half of all known pathogenic genetic variants, like the mutations that actually cause human disease, are due to single nucleotide variants, or SNVs.
So just one wrong letter.
Exactly. Furthermore, if you look at all observed human genetic variants across the board, over 96% of them are SNVs.
Wait, 96%. So
Yeah.
almost all human genetic variation comes down to a single letter change.
Yes, these are single-letter typos in a genome composed of over 3 billion letters.
Wow.
So if a patient has a severe disease caused by one single incorrect letter out of 3 billion, using a molecular scissor to chop their entire double helix in half is a massive, dangerous overreaction. It is using a sledgehammer to crack a nut.
Okay, so we bring in the base editor to be the atomic watchmaker. But here's the part that it really confused me in the source material. The base editor still uses the Cas9 protein.
It does, yes.
It uses the very protein that became world famous specifically for being a molecular scissor. So how do we actually stop it from cutting the DNA?
By chemically disarming it. Bioengineers took the natural Cas9 protein and mutated the specific catalytic domains that act as the blades of the scissors, specifically the ROVC and HNH nucleus domains.
Okay.
By mutating key amino acids in those domains, they engineered what we call a catalytically impaired Cas9. And there are two flavors of this.
Right, I remember seeing dCas9 and nCas9.
Exactly. The first is a completely dead Cas9, known as dCas9, which cannot cut either strand of the DNA at all.
It's totally blended.
Totally. The second, and far more common in modern base editing, is a nickase version, called nCas9. The nickase has one working blade and one broken blade. It can only snip one single strand of the DNA, not the whole double helix.
So it still uses its guide RNA to search the entire 3 billion letter genome. It still perfectly locks onto the exact target address, but the scissors are blunted. It's essentially just a highly targeted delivery truck.
That's exactly what it is. It parks at the target DNA and acts as a homing beacon. And fused to this deactivated Cas9 delivery truck is an entirely different payload, a totally separate enzyme called a deaminase.
And what happens when it parks? Because from what I understand, the deaminase enzyme can't just, like, reach out and edit a normal, tightly wound double helix of DNA. The letters are hidden inside the twist of the ladder.
They are. The nitrogenous bases are deeply protected within the hydrophobic core of the double helix. But the Cas9 delivery truck does something very specific to the DNA structure when it binds to its target.
Oh, does it force it open?
It does. Through a combination of molecular binding energy and structural shifts, it locally denatures the double helix. It physically pries the two strands apart over a very short distance, generating a structure we call an R loop.
An R loop.
Yeah, it essentially unzips a tiny transient bubble of single-stranded DNA.
So it forcefully pops the hood of the car, exposing the internal engine components so the devious mechanic can actually reach in and do the work.
That's a really good way to visualize it. And that exposed single-stranded bubble creates a highly localized, strictly defined editing window.
Because the enzyme is physically attached to the truck, right?
Exactly. Because the deaminase enzyme is tethered to the Cas9 protein by a short linker of amino acids, it only has a limited reach. It operates on a triphasic sequence, but it can only act within a very narrow window, typically just 4 to 8 nucleotides wide on that unzipped strand.
So if your target genetic typo falls exactly within that specific 4 to 8 base window, the deaminase can reach it and perform its chemistry.
Right. If the typo is at position 10 or position 2, it is physically inaccessible. The linker just can't stretch that far, or the DNA hasn't been unzipped in that spot.
That is just an astonishingly elegant piece of engineering. But let's get into the actual mechanic doing the work under the hood.
The chemistry.
Yes, the deep chemistry. If the Cas9 nickase is just holding the bubble open and it's not cutting the double helix apart, how does the actual letter swap happen? Like, what is that attached deaminase actually doing to the physical DNA letter to change it from a C to a T?
Well, to really grasp that, we have to zoom way in. We need to move from the macrostructure of the protein complex down to the subatomic chemistry.
Let's do it.
There are two main categories of DNA base editors. Let's start with the first. Cytosine base editors, or CBEs. Their entire job in life is to convert a cytosine-guanine base pair into a thymine-adenine base pair.
A CG to a TA.
Right. To do this, they use a naturally occurring enzyme called a cytidine diminase, most commonly one called ApoBec1 or another one called AID.
Okay, let's unpack this ApoBec1 enzyme. How exactly does it perform this alchemy? How does it look at a C and turn it into a T?
It performs a chemical reaction called hydrolytic deamination. Let's look inside the active site of the ApoBec1 enzyme. It relies on a highly conserved, zinc-dependent catalytic domain.
Zinc, like the mineral.
Exactly. This is a brilliant example of bioinorganic chemistry. Deep within the enzyme, there's a specialized pocket that tightly coordinates a single divalent zinc ion. And this zinc ion is held suspended within a specific geometric structure called a tetrahedral ligand field.
A tetrahedral ligand field. Okay, that sounds like something straight out of a physics textbook. Can you translate that for us?
Sure. Imagine a microscopic pyramid. At the center of the pyramid is the zinc atom. At 3 of the 4 corners of this pyramid, the zinc is held tightly in place by 3 specific amino acids from the enzyme itself. 1 histidine residue and 2 cysteine residues.
So it's like a tripod holding.
Basically, yeah, they form a rigid structural claw. But at the 4th corner of the pyramid, the zinc atom grabs onto a single molecule of ambient water from the surrounding cellular fluid.
So you have this microscopic claw holding a zinc atom, which is dangling a single water molecule right over the target cytosine letter on the DNA.
Yes. And here is where the catalysis happens. Near that water molecule is another crucial amino acid, a glutamate residue. This glutamate acts as a general base. It chemically reaches out and pulls one of the protons, a hydrogen atom, right off that coordinated water molecule.
Wow. And when you rip a proton off H2O, you get...
You are left with an OH minus a highly reactive nucleophilic hydroxide ion.
It essentially weaponizes a water molecule.
It really does. That newly formed hydroxide ion then physically attacks the cytosine molecule. Specifically, it attacks the carbon atom at the number 4 position of the cytosine's pyrimidine ring.
And what does that attack do?
This nucleophilic attack disrupts the chemical bonds and forces the cytosine to release its amino group. The enzyme then thoughtfully shuttles a proton back onto that leaving amino group, and it breaks off entirely, floating away harmlessly as a molecule of ammonia.
Wow, so we haven't touched the phosphodiester backbone of the DNA at all. We just lowered a zinc claw, weaponized some water, and ripped an amino group off the side of the letter C.
That's it. And by ripping off that one tiny amino group, the fundamental geometry and hydrogen bonding profile of the molecule changes completely. It is no longer cytosine. It has chemically transformed into an entirely different molecule, uracil.
But wait, uracil, we said a minute ago that cytosine base editors turn C into T, not U?
They do, ultimately. But uracil is the necessary intermediate. Once the cytosine becomes a uracil, the base editor unminds and leaves.
Okay, so it just leaves U sitting there.
Right. And the cell eventually undergoes its normal cycle of DNA replication. During replication, the cellular polymerase reads that new U, and because of its chemical shape, pairs it with an adenine and A.
Oh, I see.
And then in the next round of replication, that A is paired with a thymine, a T. So the original CG pair becomes a UG mismatch, then a UA pair, and finally, permanently, a TA pair.
That is the magic of deamination. But this brings us to the second type of editor, which I found even more fascinating in our sources. Adenine base editors, or ABEs.
Yes, ABEs.
Their job is the opposite. They convert an adenine-thymine pair into a guanine-cytosine pair. But the literature pointed out a massive roadblock when scientists first tried to build this. There was literally no known natural enzyme on Earth that could deaminate adenine in single-stranded DNA.
It was a massive hurdle. Nature had provided ApoBec enzymes to edit cytosine, mostly as a natural defense mechanism against viral infections. But nature never evolved an enzyme to deaminate adenine within DNA.
Not at all.
Well, there are enzymes that do it in RNA, but RNA has a very different chemical structure and flexibility.
So scientists hit a wall. Nature didn't build the tool, so what did? They just invented a completely new enzyme from scratch.
Essentially, yes. They engaged in a tour de force of directed evolution. They started with an E. coli bacteria enzyme called TADA, which normally only acts on transfer RNA.
Okay.
They had to force this TADA enzyme to accept DNA instead. The evolved synthetic TADA uses a very similar zinc-binding motif to what we just discussed, specifically using His57, Cis87, and Cis90 to hold the zinc atom.
Same claw.
The exact same kind of claw. In this synthetic active site, the glutamate residue helps the water molecule attack the C6 carbon of the purine ring of adenine. Ammonia is released, just like before, and the adenine is converted into a molecule called inosine.
Inosine.
Again, we're trying to get to guanine, the letter G. Why are we making inosine?
Because biology is deeply pragmatic. When a cell's DNA polymerase enzyme is chugging along the DNA track during replication and it encounters an inosine, it looks at its chemical face, looks at its hydrogen bonding patterns, and interprets it chemically as a guanine.
It just reads it as a G.
It reads I as G, so it functions perfectly as a G, pairing it with a C down the line.
Okay, I follow the chemistry, but I have a major pushback here, based on basic high school biology. The cell is incredibly smart and fiercely protective of its genetic code. DNA is not supposed to have uracil or inosine in it.
No, it's not.
Uracil is strictly an RNA letter, so if the cell is patrolling its own genome, scanning its DNA, and suddenly finds an RNA letter sitting there, a U sitting awkwardly across from an old G, alarms must be ringing.
Oh, absolutely. It's like an overzealous spell checker scanning a document written in American English and suddenly spotting a British spelling. It's going to highlight that word in bright red and immediately try to fix it back to an American C.
That is the perfect analogy, and it was the second major hurdle the bioengineers faced. Mammalian cells are packed with an enzyme called uracil DNA glycosylase, or UNG.
ONG.
Yeah. UNG is essentially that hyperactive spell checker. Its entire biological purpose is to patrol the genome, recognize uracil bases that shouldn't be there, physically cleave the bond holding the uracil to the DNA backbone, and initiate a massive cellular repair process called base excision repair.
Which would undo all our hard work.
Completely. The end goal of that repair is to revert the sequence right back to the original cytosine. If UNG has its way, your highly advanced precision base editing attempt is completely erased within hours.
So how on earth did the bioengineers outsmart a billion years of evolved cellular spell checking?
They turned to nature's own evolutionary arms races. They looked at bacteriophages, viruses that specifically hunt and infect bacteria.
Okay, what do phages have to do with this?
Well, these viruses often use uracil in their own DNA to evade bacterial immune systems, so they evolved ways to stop the spell checker. The researchers took a tiny protein derived from one of these phages, known as uracil glycosylase inhibitor, or UGI.
UGI?
Right. They took the genetic code for UGI and physically fused it to the Cas9 deaminase complex. So now the delivery truck carries a third passenger.
And what does this UGI passenger do?
UGI acts as an incredibly precise DNA mimic. It physically folds itself to look just like DNA. It latches directly onto the active site of the cell's UNG enzyme and completely binds it up.
It jams machinery.
It effectively puts a lock on the spell checker's hands. It ensures that the newly created uracil is protected from being ripped out of the genome.
It throws a blanket over the spell checker's eyes. Nothing to see here. Keep moving.
Exactly. But as clever as that is, protecting the uracil is still only half the battle. Think about the physical state of the DNA at this exact moment. You now have a massive mismatch in the double helix. One strand has the brand new U, which we want to eventually become a T. The opposite, untouched strand, still has the old G.
And U and G do not pair together. The rungs of the ladder are physically broken.
Right. And the cell's mismatch repair machinery will eventually detect the structural bulge in the ladder and force a fix. It knows one of those two letters is wrong, but the repair machinery is a blind librarian. It doesn't know which letter is the mistake.
Oh, man, so it's a 50-50 shot.
Exactly. How do the bioengineer ensure that the cell decides to fix the old strand to match the new edit, rather than reverting your new edit to match the old strand? It's a coin flip.
You would have to somehow trick the cell into believing the old pristine strand is actually the damaged one. How do you trick a system that complex?
This is exactly why we use the Nickcase version of Cas9 rather than the completely dead dCas9. The Cas9 Nickcase is programmed to specifically nick, meaning to make a tiny little cut on the phosphodiester backbone of the unedited DNA strand.
To strand with the old G.
Yes. In the rigid logic of cellular mismatch repair, if a cell finds a mismatched base pair and it sees that one of the strands nearby has a physical nick or break in it, it assumes that the broken strand is a newly synthesized, highly error-prone strand.
It thinks it's a fresh mistake.
Yes, and it assumes the unbroken strand is the pristine, ancient template. So the mismatch repair machinery sees the nick we deliberately put on the G strand. It assumes the G strand is the error, it degrades the G strand, and synthesizes a brand new strand using our newly edited U as the master template.
That is genuinely diabolical. I love it. You make the pristine edit on one side of the ladder, and then you deliberately take a knife and scratch the opposite side of the ladder just so the cell's repair crew looks at it and says, oh, clearly this scratch side must be the broken one. I'll just copy from this weird new U side.
Yeah.
And just like that, the UG mismatch becomes a UA pair, which permanently becomes a TA pair. You have officially hacked the host to finalize your own edit.
It is a masterclass in hijacking cellular machinery to do your bidding. You aren't just performing chemistry, you are manipulating biological logic.
Here's where it gets really interesting, though. The chemistry is elegant, hijacking the spell checker is clever, the nicking trick is diabolical. But our sources are very clear. These enzymes are far from perfect.
No, they're not.
We talked earlier about that editing window, that 4 to 8 nucleotide gap where the unzipped DNA is exposed. What happens if my target mutation is a single C that I need to change to a T, but sitting right next to it, completely healthy and normal, is another C, and they are both exposed in that tiny bubble?
This is the phenomenon known as bystander editing, and it is one of the biggest safety hurdles in the field. Because the deaminase enzyme is highly active and somewhat promiscuous, it doesn't just precisely hit the one nucleotide you want.
It just hits everything.
It chemically attacks whatever target bases are accessible within that exposed single-stranded bubble. If your target A is flanked by another A, the TAT-A enzyme might convert both of them to inosine.
It's collateral damage.
It is. Now, in some genetic diseases, altering the adjacent base doesn't actually change the resulting amino acid. It's a silent mutation, so the bystander edit is harmless. But in many others, that bystander edit creates an entirely new, potentially highly pathogenic mutation right next to the one you just cured.
So how do you fix a microscopic enzyme? How do you make it pickier? You can't just take a tiny wrench and tighten the catalytic claw.
You have to map the enzyme's atomic interactions in three-dimensional space and literally re-engineer its structural sequence. You have to change the amino acids to make the active site slightly narrower or change its electrostatic charge.
It's hard to do.
It's incredibly hard. To do that accurately, scientists have hit the limits of classical computing. This is where biotechnology meets quantum mechanics.
This was the source that completely blew my mind. We have a paper discussing how researchers used an IBM Qiskit 25-qubit quantum computer to simulate the exact transition states of that TAD-A 8.20 enzyme we talked about.
Yes, the quantum simulation.
I have to admit, I got lost in the physics here. Why couldn't a normal supercomputer, like the ones that predict global weather patterns or simulate galaxies, just simulate this enzyme?
It comes down to the staggering complexity of electron interactions during chemical catalysis. To accurately simulate the quantum tunneling, the proton transfers, and the exact transition states of the adenine deamination reaction, you can't just model atoms as rigid balls on sticks.
You have to go deeper.
You have to track the probabilistic behavior of the individual electrons in the active site. The minimal simulation to even begin to understand this enzyme required tracking 25 atomic orbitals. And when you factor in the critical water and ammonia molecules involved in the reaction that we discussed earlier, the model rises to 32 to 33 interacting orbitals.
And on the surface, to a layperson, tracking 33 orbitals doesn't sound like a computationally massive number.
It doesn't. But in quantum chemistry, electrons are indistinguishable fermions, and their states become heavily entangled. Trying to calculate what we call the full configuration interaction, which means mathematically calculating every single possible arrangement, repulsion, and interaction of those electrons simultaneously.
It just scales too fast.
It scales exponentially, the math explodes. For 33 active orbitals, the amount of data required to hold that state is simply intractable for classical bits of ones and zeros. The largest classical supercomputers on Earth simply cannot process the combinatorial explosion of the wave functions.
So how does a quantum computer, with only 25 qubits, handle something a supercomputer can't?
Because a quantum computer doesn't simulate the quantum mechanics, it is quantum mechanics. They utilized an algorithm called the Variational Quantum Eigensolver, or VQE.
VQE, okay.
In quantum computing, you can map the actual state of these fermionic spin orbitals directly onto the physical quantum states of the qubits themselves. The researchers used a mathematical bridge called the Jordan-Wigner transformation.
Which does what, exactly?
This translates the complex fermionic operators, the heavy math describing the biological electrons, into Pauli matrices, which are physical operations the qubits can directly execute.
Let me make sure I'm wrapping my head around this.
Yeah.
The qubits inside the IBM machine are literally mimicking the physical behavior of the electrons inside the biological enzyme's active site.
Exactly. And by mapping it this way, they were able to calculate the exact, highly complex energy landscape of the chemical reaction. They watched in virtual quantum space as that specific glutamate residue, Glu59, acted as a general base to nucleophilically attack the C6 carbon.
Wow.
They could visualize the exact atomic distances, the electrostatic repulsions, and the energy barriers the molecule has to overcome to swap that letter.
Okay, so you run this incredibly dense quantum simulation. You map the energy landscape. How does that help a biologist standing in a wet lab actually build a better base editor to stop those bystander edits? How does quantum math become a physical cure?
Understanding the energy landscape and the spatial constraints of the active site tells you exactly which structural pillars of the enzyme you can mutate without destroying its function. It guides directed evolution.
Okay.
The researchers knew they needed an editor that was smaller, highly efficient, but incredibly specific. Now, traditionally, cytosine base editors, the ones that use the natural ApoBec enzymes, are quite large and have a much higher propensity for those Cas-independent off-target effects.
Which means what exactly?
They are known to sometimes randomly float around and edit ambient DNA or RNA floating in the cell, completely independently of the Cas9 guide.
So they are a bit too trigger-happy. They deaminate things they aren't supposed to touch.
Precisely. So researchers did something remarkable, guided by these deep structural insights. They took the highly evolved adenine editor Tadaive 80, which we already knew was very stable and had incredibly low off-target activity, and they forced it to evolve into a cytosine editor.
Wait, hold on. I want to make sure I heard that right. They took the machine, they explicitly designed to turn A to G, and they rewired its core logic to turn C to T instead.
Yes. They used a grueling technique called phage-assisted continuous evolution, or PACE-E, and its non-continuous variant PANS.
How does that work? How do you force an enzyme to change its target?
You link the survival of a virus to the success of the enzyme. They created an artificial system where bacteriophage viruses can only survive and reproduce in a bacterial host if they successfully mutate the TADA enzyme payload to deaminate cytosine instead of adenine.
So if it doesn't mutate, it dies.
If the enzyme fails, the virus dies. Over many generations of rapid, forced, intense evolution, the enzyme accumulated specific mutations in its DNA-binding residues. It chemically altered its own selectivity pocket to strongly favor deoxycytidine.
That is unbelievable. It's essentially artificial natural selection running at hyperspeed in a test tube. So they created a totally new, completely synthetic class of cytosine editor derived entirely from the adenine editor framework.
They are known as TADCBEs. And because they are built on that robust Tatada chassis rather than the wild ApoBec chassis, they are physically smaller, they have vastly reduced off-target RNA and DNA bystander editing, and yet they maintain incredibly high on-target activity.
That's amazing.
And the structural insights from these kinds of evolutionary leaps even led to the creation of dual editors.
Dual editors, like performing both chemical swaps at the exact same time.
Yes, tools like the ANCB Emacs, it takes a cytidine deaminase and an adenosine deaminase and fuses both of them to a single Cas9 nickase.
Oh, wow. So you have three things attached now.
Yeah. So imagine a patient has a highly complex pathogenic mutation where you need to change a C to a T and right next to it an A to a G. Before, you might have to run two separate gene therapies. Now, this single dual-headed machine can park on the DNA, unzip the R loop, and perform both distinct chemical conversions simultaneously within the exact same editing window.
That is molecular engineering at its absolute pinnacle. It's hard to even fathom the precision. But taking a step back here, even with perfect, quantum-engineered, dual-wielding, non-bystander base editors, we are still fundamentally talking about making a permanent, irreversible change to the human genome.
We are. It's a big deal.
And that scares people. What if we get it wrong? What if the patient has a reaction? What if you don't want to chisel a permanent change into the master stone tablet of the cell's DNA? What if you just want to send out a revised biological memo that eventually goes in the trash once the patient gets better?
That is exactly where the RNA alternative comes in, and it's a massive subfield in genetic medicine right now. To use your analogy, DNA is the hard drive, it is permanent, archival storage, RNA is the RAM, it is temporary, transient, and constantly being degraded and refreshed by the cell.
So it's not forever.
Right. If you edit the messenger RNA transcript, the mRNA, instead of the genomic DNA, you can completely alter the final protein the cell produces, curing the disease phenotype without ever touching the patient's permanent genetic code. It's a temporary, dose-dependent, and fully reversible fix.
So how does RNA editing differ mechanically from the DNA-based editing we just spent 20 minutes dissecting? Are we still using our trusty delivery truck, Cas9?
Not necessarily, and sometimes not at all. There are CRISPR-based systems for RNA, like Rescue, which performs CDU editing, and Repair, which performs A to I editing.
Okay.
But they don't use Cas9. They utilize a completely different Cas protein called Cas13b, which specifically targets and binds to single-stranded RNA instead of double-stranded DNA. But the truly elegant approach to RNA editing doesn't require delivering a massive foreign Cas protein into the body at all. You can just use antisense oligonucleotides, or ASOs.
ASOs. What exactly are those structurally?
They are incredibly short, highly stable synthetic strands of RNA. You engineer them in a lab to perfectly complement and bind to the specific target mRNA sequence in the patient's cells that you want to edit.
Like a magnet.
Like a perfect complementary puzzle piece. When you inject the ASO, it floats around until it finds its target mRNA transcript and binds to it, creating a small stretch of double-stranded RNA. Now, our cells naturally contain vast reserves of enzymes called ADARs, adenosine deaminases, acting on RNA.
Okay, natural editing enzyme.
Right. These natural ADARs patrol the cell looking for double-stranded RNA, and when they find it, they naturally perform A to I editing on them. So the synthetic ASO acts as a molecular guide. It forcefully recruits the body's own natural endogenous ADAR enzymes to the exact spot on the disease-causing mRNA that you want edited.
Oh, that is incredibly clever. You don't have to worry about the immune system rejecting a massive bacterial Cas9 protein because you aren't delivering a protein. You just deliver a tiny piece of synthetic guide RNA, and it recruits the cell's own natural workforce to do the heavy lifting.
It is highly efficient for delivery and massively lowers the risk of immune toxicity. But the challenge with RNA editing is that because you are relying on the body's natural systems, you are subject to the body's natural rules.
Ah, the fine print.
Yeah. The natural RNA editing pathways are incredibly complex and heavily regulated by biology. There are natural cellular roadblocks designed to stop runaway editing. And this brings us to a fascinating, highly specific physiological study from our sources, dating back to 2001, on an obscure enzyme called ARCD1.
Yes, I wanted to ask about this because it seemed like a very deep cut into basic physiology compared to the quantum computing stuff. What exactly is ARCD1?
ARCD1 stands for ApoBec1-related cytidine deaminase 1. Genetically, it's encoded on human chromosome 6p21.1. Structurally, it has the exact same standard zinc-dependent cytidine deaminase active site we discussed earlier with the tetrahedral ligand field.
So it's an editor.
You'd think so. But when researchers in 2001 isolated it, they made a baffling discovery. When they tested ARCD1 in a purified test tube, it possessed strong cytidine deaminase activity. But when they put it inside a living cell, it could not catalyze the CDU editing of ApoB mRNA on its own.
Even though it has the right claw.
Right. And ApoB mRNA is the classic standard natural RNA editing target in the human liver and intestines.
Wait, I'm confused. It's an editing enzyme that has a working active site, but it fundamentally cannot edit its own primary target inside a cell. What is the point of it then?
This was the exact question that stumped researchers and raised fundamental questions about biological regulation. Through further study, they discovered that ARCD1 actually acts as a dominant negative inhibitor.
A dominant negative inhibitor.
Its evolutionary purpose is not to edit RNA. Its purpose is to physically stop other enzymes from editing RNA.
Why on earth does the body spend cellular energy building an enzyme whose sole job is to tackle another enzyme and stop it from working?
Because RNA editing is so incredibly powerful, the body needs a highly sensitive, universally expressed volume knob to turn it up or down so the cell doesn't just dissolve into chaos. To prove this, the researchers performed highly rigorous experiments using a specific line of cells called McArdle 777 rat hepatoma cells.
Hepatoma, so liver cells.
Right. They used a recombinant adenovirus vector to artificially force these liver cancer cells to overexpress ARCD1. What they found was that the more ARCD1 they added, the less natural RNA editing occurred. ARCD1 physically seeks out and binds to the true active editing enzyme, ApoBec1, and its necessary complementation factor, an accessory protein called ACF.
So it literally physically tackles the active editor complex.
Yes. Through a biochemical technique called far western analysis, which is a method used to detect specific protein interactions, they proved that ARCD1 heterodimerizes with ApoBec1. It locks onto it, creating a deactivated pair.
And it's just everywhere.
It is ubiquitously expressed throughout the body with particularly high levels in skeletal and cardiac muscle, and localizes in both the cell nucleus and the cytoplasm. It acts as a universal, ever-present dampener. It ensures that the highly active ApoBec1 doesn't just run wild and indiscriminately edit every single piece of mRNA floating in the cell, which would cause massive protein dysfunction and cell death.
Okay, that makes sense as a natural biological fail-safe.
Uh-huh.
But bringing this back to our clinicians and biotech investors,
Yeah.
if our bodies are naturally pumped full of these ubiquitous dampener proteins like ARCD1 that actively seek out and shut down RNA editing complexes, aren't these biotech companies just throwing billions of dollars into therapeutic ASOs that our own biology is fundamentally designed to neutralize?
That is the multi-billion-dollar question, and it is exactly why understanding basic physiology, like the ARCD1 study, is so critical for modern bioengineers.
You have to know what you're fighting.
Exactly. If you want to perform therapeutic RNA editing in a human liver, you aren't just operating in a vacuum or a clean test tube. You are entering a highly combative, heavily regulated cellular environment teeming with natural inhibitors. Your synthetic ASOs, or your delivered Cas13 editors, have to be engineered to either massively outcompete these natural roadblocks through high binding affinity, or be designed in a way that navigates entirely around them. The biology fights back.
Which is why the permanent DNA fix is still the holy grail for so many companies.
Huh?
So RNA editing gives us a temporary, heavily regulated fix. Base editors give us permanent, highly precise single-letter fixes in the hard drive.
Right.
But I have another scenario.
Yep.
What if a patient's genetic disease isn't caused by a single wrong letter? What if they're missing an entire word, or a whole paragraph of genetic code is backward? I'm thinking of massive insertions or deletions.
Yeah.
You fundamentally can't fix a missing paragraph with a simple pencil and an eraser.
Oh.
You can't just change an A to a G.
No, you can't.
You need a word processor. You need to be able to highlight a giant section, hit delete, and type in a whole new sentence.
And that profound limitation brings us to the most complex, massive tool in the entire precision editing arsenal, prime editing.
Prime edit.
If base editing is the pencil, prime editing is true search and replace. It can perform insertions of new DNA, deletions of massive chunks of DNA, and all 12 possible single base substitutions, all without ever relying on the catastrophic double-strand breaks of traditional CRISPR.
Okay, let's unpack prime editing.
Hmm.
Because mechanically, this sounds like black magic. How do you build a word processor for DNA? I assume you can't use the deaminase enzyme anymore, right? Because a deaminase can only tweak a letter. It can't build new ones.
Right. The structural architecture of a prime editor, or PE, is fundamentally different from a base editor. It still utilizes the trusty Cas9 nickase delivery trick, so it is only safely snipping one strand of the DNA.
Right.
But instead of a deaminase being tethered to it, the Cas9 is fused to a massive, highly complex engineered enzyme called a reverse transcriptase.
Reverse transcriptase. Wait, that's the enzyme retrovirus is like HIV used to hijack cells, right? It takes an RNA sequence and turns it into permanent DNA.
Yes, exactly. It reads a temporary RNA template and physically synthesizes a brand new permanent strand of complementary DNA based on that template. To direct this entire word processing event, prime editing uses a highly specialized, very long guide RNA called a pegRNA prime editing guide RNA.
So it's not just a normal guide.
No, the pegRNA is an absolute masterpiece of molecular design. It doesn't just have the targeting sequence to find the right address on the genome like classical CRISPR. It extends much further and contains two crucial extra domains, a primer binding site, or PBS, and a template region containing the actual new genetic code you want to insert into the patient.
So the guide RNA doesn't just give the address, it actually carries the new blueprint, the new paragraph, right there on its back.
Yeah.
How does the physical insertion actually happen at the DNA level?
The mechanism is a breathtaking piece of molecular choreography. The Cas9 nickase parks at the target and cuts just one strand of the DNA. This creates a loose flapping end of cut DNA.
Right.
The primer binding site on the pegRNA reaches out and hybridizes it, chemically pairs and binds to that loose DNA flap. This anchors the RNA solidly to the DNA strand. Then, the reverse transcriptase enzyme kicks into gear.
And starts typing.
It grabs the end of the nicked DNA flap, looks at the template region of the pegRNA right next to it, and begins synthesizing brand new nucleotides, physically extending the DNA strand, typing the new genetic code directly onto the end of the flap.
It is literally 3D printing fresh, corrected DNA directly into the genome sequence, letter by letter.
Precisely. And once it finishes typing out the new DNA, you have this chaotic intermediate state. The DNA ladder now has a long, loose flap dangling off of it, containing the new edits, while the original unedited strand is still attached underneath it.
That sounds unstable.
It forms a structure called a 3' flap. The cell's natural endonucleases enzymes that clean up loose DNA ends will eventually snip away the old, unedited flap, and the new edited flap chemically seals itself into the genome.
But hold on, let me get this straight. We just talked about the mismatch repair system with base editing. If you successfully 3D print a brand new paragraph on one side of the DNA ladder, it absolutely will not match the unedited sequence on the other side of the ladder. Doesn't the cell spell checker freak out and just erase the whole thing you just printed?
An excellent catch, and that exact problem is exactly how prime editing had to evolve from its first prototype, PE1, to the advanced PE3 architecture used today.
PE3, right?
In the PE3 architecture, bioengineers utilize a standard second guide RNA to direct the Cas9 complex to make an additional nick on the unedited strand, far enough away to safely avoid causing a double-strand break, but close enough to trigger the cell's mismatch repair machinery.
Oh, the same trick.
Just like the trick we discussed in base editing. The cell sees the nick on the unedited strand, structurally assumes it's the broken one, degrades it, and uses the newly prime-edited strand with the inserted paragraph to copy the new sequence over to the opposite side.
So it makes the massive insertion permanent on both sides of the helix. That is staggering. One of the clinical sources we reviewed highlights Tay-Sachs disease as a prime editing proof of concept.
Yes, Tay-Sachs is a great example.
Tay-Sachs is a devastating fatal neurodegenerative disorder in children, often caused by a 4-base pair insertion in the XXA gene. Standard base editing fundamentally cannot fix that because it's not a single letter change. But prime editing can just search for that 4-base insertion, highlight it, and delete it perfectly, restoring the gene.
Exactly. Prime editing opens the door to correcting massive, complex structural mutations that were entirely out of reach for molecular pencils.
But I'm going to push back here on the utility of base editing then. If prime editing is this incredibly powerful word processor, if it can do everything a base editor can do, plus massive insertions, plus deletions, plus all 12 possible base conversions, why do biotech companies still use base editing at all? Why not just throw the pencil in the trash and use the word processor for every single disease?
If we look at the reality of clinical application and manufacturing, the answer comes down to two major, heavily restrictive factors: biological efficiency and sheer physical size.
Okay, let's talk efficiency.
Prime editing is incredibly versatile, but that versatility comes at a severe cost of efficiency. The mechanism we just described is highly complex, relying on multiple delicate hybridization steps, RNA anchoring, and reverse transcription. It has a lot of moving parts that can fail.
More parts, more problems.
Right. In many human cell types, if the target mutation is just a single letter and it falls perfectly within the strict 4 to 8 base window of a base editor, the base editor will achieve significantly higher editing efficiencies, often with far fewer unintended indel byproducts than a prime editor attempting the exact same edit.
Okay, so if a simple, reliable pencil eraser works for the typo, use the eraser. Don't boot up the entire slow, complicated word processor just to delete one letter.
Precisely. And the second, arguably more critical factor is size. Prime editors are physically massive proteins. The genetic sequence required to code for a fusion of Cas9 to a reverse transcriptase creates a payload that can be up to 7.0 kilobases long.
And base editors are smaller.
Base editors are significantly smaller, usually around 5.0 to 5.5 kilobases. And physical size is the absolute ultimate bottleneck when we transition from talking about cool biochemistry in a petri dish to turning these molecular machines into actual medicine you can sell and inject into a patient.
Because you actually have to get these massive proteins inside a living human cell, inside a living human organ. Which brings us to the final and perhaps most crucial piece of the puzzle for the clinicians and business people listening: delivery.
The delivery challenge.
Right. We can build the most elegant, quantum-engineered, dual-wielding prime editor in the world, but if we can't get it inside a patient's liver cells, it's completely useless.
Delivery is the absolute crucible of modern gene therapy. Historically, the most popular delivery vehicles for gene editing are viral vectors, specifically a highly engineered, harmless virus called an adeno-associated virus, or AAV.
AAVs are fantastic at homing in on human cells and injecting their genetic payload.
Yeah.
But they have a strict, unforgiving physical packaging limit of about 4.7 kilobases of DNA.
And you just explicitly said base editors are 5.5 kilobases and prime editors are 7.0 kilobases. They literally physically do not fit inside the delivery truck. They absolutely do not. Researchers have tried highly complex workarounds. They have tried physically splitting the editor proteins in half, packaging them into 2 separate AAV viruses, injecting both, and hoping they reassemble themselves using special interlocking protein domains inside the cell.
That sounds like a long shot.
It drastically lowers the therapeutic efficiency because you are relying on statistical luck, hoping both viruses happen to infect the exact same cell at the exact same time. Furthermore, AAVs, because they are viral shells, can trigger significant immune responses.
Right, your body recognizes it as a virus.
Exactly. Patients often develop neutralizing antibodies after the first dose, meaning you can usually only ever dose a patient once. If the first dose doesn't cure them, you can't try again.
So what is the business solution here? If the viruses are fundamentally too small, how are these multi-billion-dollar biotech companies actually getting these massive cures into human clinical trials right now?
The entire field is executing a massive pivot toward non-viral delivery systems, specifically focusing on lipid nanoparticles, or LNPs.
LNPs. That is the exact same technology that was used to deliver the mRNA COVID-19 vaccines globally, right?
Exactly. LNPs are microscopic, highly engineered spheres of fat that encapsulate the messenger RNA encoding the base editor protein, along with the guide RNA. LNPs can carry vastly larger payloads than the cramped 4.7 kilobase limit of AAVs.
So the size limit is gone.
Right. A 7.0 kilobase prime editor fits comfortably inside an LNP. Furthermore, because LNPs are just lipid shells without viral proteins, they don't trigger the same severe adaptive immune responses. This means, theoretically, they could be redosed if a patient needs a booster edit.
Wow.
And from a pure manufacturing and business standpoint, synthesizing lipids and mRNA in a chemical plant is significantly cheaper, faster, and more scalable than growing millions of batches of live viruses in massive bioreactors.
This is exactly where the capital is flowing. If you are a business strategist looking at the gene editing landscape, the massive pivot from viral manufacturing facilities to LNP formulation technology is the major tectonic shift of the decade.
Absolutely.
And we are seeing this exact LNP strategy play out in the clinical pipeline right now. Let's look at the actual human clinical trials from our sources, because this is where the science hits reality. Beam Therapeutics, one of the original pioneers in this space founded by David Liu, is advancing a therapy called Beam 301.
Beam 301 is a fascinating and highly critical program. It is an in vivo LNP base editor, meaning the editing happens inside the living body, specifically targeting the liver.
And what's it targeting?
Its goal is to correct the R83C mutation. This is a severe single-point mutation that causes glycogen storage disease, type 1A, a rare metabolic disorder where the liver cannot regulate blood sugar, requiring patients to consume specialized cornstarch around the clock just to survive.
Just to keep their blood sugar up.
Yeah. By using an LNT to deliver the base editor directly to the patient's liver cells, they aim to permanently rewrite that single mutated letter, restoring the liver's natural ability to process glycogen.
No viruses, no double-strand breaks, just a microscopic droplet of fat carrying a molecular pencil into the liver to erase a disease. And then there's Verve Therapeutics, which is taking a totally different approach. They aren't going after ultra-rare diseases. They're going after cardiovascular disease, one of the biggest killers on Earth.
Verve is tackling heart disease at the genetic level. They are developing VERVE-101 and VRV-102. These therapies use specially targeted GalNAc LNPs.
GalNAc, what's that?
GalNAc is a specific chemical sugar tag added to the outside of the lipid nanoparticle that acts like a zip code, ensuring the LNP is exclusively absorbed by specific receptors on liver cells. But their goal is not to correct a mutated gene. They are using an adenine base editor to introduce a deliberate stop codon to turn off the PCSK9 gene in patients with heterozygous familial hypercholesterolemia.
So for breaking it on purpose.
Precisely. PCSK9 is a gene that naturally impedes the liver from clearing bad LDL cholesterol. By turning off PCSK9 with a single, highly precise base change, they aim to drastically and permanently lower LDL cholesterol levels.
That is essentially a permanent genetic statin drug administered as a single lifelong treatment. The business implications for the pharmaceutical industry of replacing daily statin pills with a one-time genetic edit are astronomical. And then we also have Correct Sequence working on severe blood disorders.
Yes. Correct Sequence is utilizing base editing ex vivo, meaning they take hematopoietic stem cells completely out of the patient's body, edit them safely in a laboratory clean room, verify the edit, and then infuse them back into the patient's bone marrow.
So it happens entirely outside the body.
Right. They are advancing CS101 for beta thalassemia, a severe blood disorder. Their base editor targets the regulatory regions of the hemoglobin genes. By making precise single base edits, they reactivate the production of fetal hemoglobin, compensating for the defective adult hemoglobin.
That's brilliant.
And crucial early clinical data shows faster activation of fetal hemoglobin and quicker patient recovery times compared to traditional CRISPR therapies that cut the DNA to achieve the same result.
And finally, Base Therapeutics is taking this technology directly into the fight against cancer with NK510. They're using their proprietary AccuBase editor to genetically engineer natural killer immune cells to actively hunt solid tumors. By using base editors instead of classical CRISPR, they entirely avoid the severe toxicity and exhaustion caused by creating double-strand breaks in the engineered immune cells before putting them back into the patient.
It is truly incredible how fast this entire field has moved.
It really is. The foundational base editing papers from David Liu's lab were only published in 2016, and here we are in 2026, putting these tools into human patients to cure heart disease, blood disorders, and cancer.
It is a remarkable testament to the convergence of multiple disparate fields. You have quantum chemistry mapping the electrons, directed viral evolution building the enzymes, RNA biology teaching us about natural cellular dampeners, and nanotechnology providing the lipid delivery trucks, all culminating in actual tangible cures.
So to briefly recap the incredible journey we've taken today, we started with the realization that molecular scissors are simply too dangerous and heavy-handed for delicate single-letter point mutations. We upgraded to the precision of the molecular pencil, the base editor. We zoomed all the way into the subatomic active site, watching a microscopic claw of zinc and weaponized water rip amino groups off cytosine and adenine.
We covered a lot of ground.
We really did. We saw how scientists used 25-qubit quantum computers to map those atomic energy landscapes to rapidly evolve entirely new synthetic editors that don't make deadly bystander mistakes. We explored the transient, highly regulated realm of RNA editing, learning how natural cellular dampeners like ARCD1 are deployed to keep the body in check.
And then prime editing.
Right. We expanded the toolkit from the pencil to the word processor with prime editing, achieving true search and replace for massive genetic insertions. And finally, we packed these massive, intricate protein machines into lipid nanoparticles to deliver them directly to patients, bypassing viral limitations to cure everything from hypercholesterolemia to glycogen storage disease.
It is a profound narrative of human ingenuity taking control of biology. But looking forward, based on the absolute bleeding edge of our sources, the implications of these tools expand even further.
Where does it go from here?
As we develop highly multiplexed base editors and advanced structural tools like TwinPE, which utilizes two massive prime editors simultaneously acting on opposite strands of DNA to insert staggering 10 kilobase sequences without a single double-strand break, We are crossing a massive philosophical and biological threshold. We are blurring the fundamental line between merely editing a genome and entirely synthesizing a brand new one from within the living cell itself.
And that leaves us with a final, provocative thought to mull over for everyone listening. Our sources detail the rise of incredibly advanced computational AI prediction tools, models like BEFF and BDOFF, that use deep learning to perfectly map and predict off-target editing effects before a drug is ever synthesized.
Right.
What happens in the next 5 years when these predictive AI models become so perfectly integrated with our quantum-engineered base editors that we can reliably and safely edit highly complex polygenic traits? When we are no longer just fixing a single typo causing a rare liver disease, but simultaneously, flawlessly, rewriting hundreds of distinct letters across the entire genome without a single unintended off-target effect. The power to completely rewrite the blueprint of life is no longer theoretical, and it is no longer 50 years away. It is sitting in clinical trials right now. Thank you for joining us on this deep dive. Keep exploring, keep questioning the mechanics of the world around you, and we will see you next time.